Hello, everyone. My name is Saburo MATSUI, Emeritus Professor of Kyoto University. I taught at Global Environmental Studies of Graduate School of the university. My background is sanitary engineering. Today, I am very much delighted to give my lecture to you, excellent participants, who are concerned sanitation business to help the people who do not access proper sanitation in the world. We are still in the cage of COVID-19 pandemic. People realize sanitation is the essential work, not to spread the virus. I hope my approach may provide you an example of the sanitation solutions. This slide shows the content of keywords. In my presentation, I would like to speak the following keywords: organic farming, phosphate limit, pesticide misuse dangers, human and ecosystem health, plant and human microorganisms, bacterial ecology, plant growth promoting rhizobacteria, PGPR, Hyper-Thermophilic composting, ecological sanitation, urine feces separation toilet, Bacillus streptomyces, and so on, bacteria. And then probiotic environmental agriculture. What is organic farming? Many health-conscious consumers are looking for organic food and environment. The answer is organic farming. US Commission say it is an agricultural method that aims to produce food using natural substances and processes, which tends to have a limited environmental impact as it encourages the responsible use of energy and natural resources, the maintenance of biodiversity, presentation of regional ecological balances, and enhancement of soil fertility. How to conduct organic farming? My answer is the probiotic environmental agriculture that promotes production and use of high-quality compost from variety of organic waste by new composting technologies. An example, hyper-thermophilic composting. High-quality compost must contain nutrients that replace chemical fertilizer and beneficial microorganisms that replace pesticides. The probiotic environmental agriculture also promotes uses of beneficial bacteria of many single species that can promote growth of plants and suppress harmful bacteria, fungi, nematodes, and insects. This slide shows you what is organic fertilizer. High-quality compost must contain essential elements. First group, for plants and human. Basic elements are carbon, hydrogen, oxygen, three major nutrient elements, nitrogen, phosphorus, and potassium. Following major elements, calcium, magnesium, sulfur. Micro essential elements are chlorine, iron, zinc, copper, boron, manganese, nickel, molybdenum. These together 17 elements. In addition to that, some plants need, such as sodium and silicate, altogether 19 elements, so that plants can survive by 19 elements. However, in addition to those 19 elements, humans need other essential elements, such as fluorine, iodine, chromium, selenium, vanadium, copper, and aluminum, and bromine. Altogether, 27 elements, which humans must obtain through food, which means through plants. Plants must take 27 elements from soil that give us healthy food. But the hazard elements that are the arsenic, cadmium, and mercury, and lead. Those harmful substances are not necessary for both plants and human. Okay, this is the first group of elements. The second group, soil must keep humus, humic acid, and fulvic acid. Today, I do not have time to explain about this. The third group, that is mutualistic bacteria with plants. So the good compost must contain three elements, three major element groups. This slide shows major phosphate rock reserving countries. You can see China, USA, Morocco, Russia, Tunisia, and others. It is predicted that economically mineable years are only 90 years. We have to find measures to this problem, which is discussed in the following slide. Figure 1. This figure showed sustainable phosphorus supply and demand measures for meeting long-term future global food demand. The ordinate indicate phosphorus annual production in the world. The horizontal axis indicates years. Up to the year 2019, the phosphorus demand and supply were met, but Cordell et al. Predict if we continue phosphorus demand in the line of business as usual, we may face a very serious conditions. That means we'll face the supply and consumption gap that will grow. We have to start any measures to fill up the gap. On the right hand, there are many programs of measures indicated. I would like to emphasize today reuse measures such as food residue, crop residue, human excreta, and animal dungs. Those must be reused. How to reuse those residues? The answer is a reuse of the form of compost. This slide showed the scope of this lecture. In order to discuss human and ecosystem health relating to human urine and feces fertilizer, we need a wide scope. We see a phylogenetic system in the center, origin of life on the earth. In the upper left hand, human, fungi, yeast, plant, nematode, and insect that are groups of domain eukaryotes. In the lower left hand, we see methane bacteria and thermophilic bacteria that are domain archaea. In the other corners, we see many domains of eubacteria groups, such as phylum Firmicutes, including Bacillus and Lactobacillus, and then phylum Bacteroides, phylum Planktomycetes, phylum Actinobacteria, phylum Cyanobacteria, and phylum Proteobacteria that include subgroups alpha, beta, gamma, so on. I am going to talk relations among human, plant, nematode, insect, fungi, and bacteria in the following slide. This slide showed the abuse of pesticides and production of resistant organisms against pesticides. That is, we face the vicious cycle of chemical pesticides. 2 million tons per year of pesticides are utilized. Herbicide 47.5%, insecticide 29.5%, fungicides 17.5%, other pesticides 5.5%. The top 10 pesticide consuming countries are China, USA, Argentina, Thailand, Brazil, Italy, France, Canada, Japan, and India. The year 2020, the global pesticide use will increase up to 3.5 million tons. This slide shows pictures of fungi disease. Many food plants are infected some groups of fungi. We developed many types of chemical fungicide. Many of fungicide created anti-fungicide fungi. How to cope with the vicious cycle? This slide shows, again, fungi and oocyte disease of plants. Plants are infected by many fungi and oocytes. Symptoms are different. In the left, Jatropha trees is infected by fungus phylum Ascomycota fusarium species. In the center upper picture, fusarium infected cucumber showing vines wilt. In the center lower picture, phylum Basidomycota Rhizoctonia solani shows dumping off of tomato. In the right lower corner, oocyst Erythricilum, that is a zygote of ovum of protozoan, infected Chinese cabbage that shows the disease yellowing blight. This slide shows bacterial diseases of plants. Plants are infected by many bacteria. Symptoms are different. In the center, the upper picture shows soft rot of tomato by gamma proteobacteria Ervina carotovora subspecies. In the center, lower picture, apple tree roots are infected by alpha proteobacteria Rhizobium radiobacter. In the left lower corner, a symptom canker of citrus infected by gamma proteobacteria Xanthomonas citri. In the right corner, rice develops a disease of seedling blight of the rice by beta proteobacteria Burkholderia plantari, and another disease of grain rot of rice by Burkholderia gourmet. This slide shows nematode disease. Root rot disease in the left. Resin nematode disease in the right are shown. Now, I would like to go deeper to a bacteria business. The table shows bacteria, phyla, and the number of bacteria species identified, which is more than 10,000. There are 30 phyla in the domain bacteria identified. It is very good. The classification of phyla according to alphabetic, starting from acidobacteria, actinobacteria, aquifer, and then in the end, Vercomicrobia. And you can see number 1, acidobacteria, number 2, actinobacteria, that had 20, number 2 actinobacteria that has 20,000 species. Number 5 bacteroides that has 500 species. Number 9 chloroflexi that has not much. Number 12 cyanobacteria that had 2,654 species. Number 14 Deinococcus sams. Number 18 Firmicute that had 2,000 species. Number 25 Proteobacteria that had 3,000 species. Number 25 Spirochetes. Those are the major bacteria. Later in my lecture, you'll see again. Those phyla that have a large number of species are playing important bacteria ecology and among human and plant and microorganisms. This slide shows a brief image of a plant. Plant grow by photosynthesis. Plant requires sunlight, carbon dioxide, water, and nutrient elements. In soil, plants develop roots by which they take up nutrients. Around the roots, plants organize a special zone called rhizosphere, where many microorganisms work for plants and attack plants. I would like to focus on the function of rhizosphere microorganisms. This slide shows a plant physiology. Plant exercise photosynthesis in leaves and continue to grow. They build the product of photosynthesis that are carbohydrates, amino acid, oils, and others in leaves. It is very important to understand their physiology. This is very important, about 10% of the product of photosynthesis, they transport it through sieve cells and discharge it from roots to the nearby soils. The discharge is called an exudate. Exudates include carbohydrates, amino acid, citric acid, malic acid, acetic acid, and so on. At the tip of roots, there is a root cap that discharges a mucigel, they call mucigel, that is mainly a pectin with exudates. These discharges are aiming at dissolving minerals nearby soils. However, it is inevitably attracting good and bad bacteria, good and bad fungi, and nematodes around roots. This slide shows plant and bacteria symbiosis and endophytes. On the leaves and at the surface of the roots, many bacteria, fungi, and nematodes are attracted. Among bacteria and fungi, good bacteria and fungi are involved in a part of plants. They are symbiotic and called endophytes. Symbiotic bacteria produce plant hormones to stimulate plant growth. A rhizosphere is very important where commensal bacteria and fungi live outside of roots. Shallow soil keeps oxygen, aerobic bacteria grow. Ammonia oxidation occurs, while deep soil that keeps anaerobic bacteria, nitrate sulfate reductions occur by bacteria. Methane is also produced. This figure shows bacterial diversity in bulk soil, in rhizosphere soil, around roots, and at seeds. This picture is so important. In bulk soil, fungi live much more than bacteria in both the number and the amount. It is very important to understand microbial ecology of soil. In bulk soil, major bacteria are phyla number 24 proteobacteria, subgroups gamma proteo, beta proteo, and alpha proteobacteria, followed by number 5 bacteroidetes, number 2 actinobacteria, and number 18 firmicutes. Altogether, 5 groups are key bacteria around roots. In rhizosphere soil, those 6 groups are balanced. Those 6 groups are balanced. At roots, alpha proteobacteria is not playing. It is interesting to see gamma proteobacteria and actinobacteria are playing something with seeds. This slide shows plant hormones that are produced by the PGPR, plant growth promoting rhizosphere bacteria. Plants produce themselves plant hormone, of course, in order to regulate the growth of leaves, roots, flowering, and fruiting. They are regulating plant life just as hormones for humans. Major hormones are auxin, such as indole 3-acetic acid, IAA, cytokinins, CK, abscisic acid, ABA, and gibberellin, GB. PGPR produce those plant hormones and stimulate plant growth. This is very interesting. This is an element of a symbiotic. More than plant hormone production, they produce many types of antibiotics and enzymes. Examples are actinomycetes produce many types of antibiotics and enzymes. One key enzyme is chitinases that dissolve the cell walls of fungi and nematodes. This is very important point. The chitinase produced by actinomycetes bacteria, chitinases are really good killing fungi and nematodes. This table 7 shows PGPR bacteria are symbiotic, most of them symbiotic, or sometimes parasitic, or sometimes pathogenic to plant, also pathogenic to human, indicating functions of plant hormones and enzyme chitinase, antibiotics, and human pathogenics. Actinobacteria, starting from upper to down, number 2, actinobacteria, many groups produce plant hormones, antibiotics, and chitinases. That is a strong enzyme that kills fungi and nematodes. Flavobacterium or bacteroidetes produce a plant hormone, but it is sometimes bacteria pathogenic. In firmicutes, such as Bacillus thuringiensis, produce insect killer proteins. Bacillus subtilis produce plant hormones and insect killer proteins. Alpha proteobacteria, Agrobacterium is plant pathogenic, but Brady rhizobium is symbiotic with soybean giving nitrogen fixing. Beta proteobacteria, alkali genes produce a plant hormone and does denitrification. Gamma proteobacteria, azotobacter is nitrogen fixing. Other bacteria produce plant hormone, but they are human pathogenic. It is important how to utilize those beneficial bacteria or plants. Okay, change the topic a little bit. The slide shows human and intestinal bacteria interaction. Human and intestinal bacteria is a key research area to better understand human health and longevity. The medical science of human immune system is rapidly developing in the interaction. Every day we are introducing food with bacteria in our gut. There is a dynamic equilibrium condition between human digestion and the gut bacteria. When we introduce good bacteria with the food, we can maintain the good equilibrium condition. In order to maintain such a good condition, we introduce good live bacteria that we call a probiotic manner. Our feces consists of 1/3 intestinal bacteria, 1/3 undigested food, 1/3 dead intestinal cells. This table shows bacterial phyla, genera, and possible species in human intestine. Human intestinal environment is very unique and semi-anaerobic, so that specially adapted bacteria are found. There are mainly number 11 phyla. 37 species are identified. But more than 10,000 species may live in human intestine, depending on individual conditions. Major bacteria are number 2, actinobacteria, number 5, bacteroides, number 18, firmicutes, and number 24, proteobacteria. There are methane bacteria of archaea live in our gut and produce the gas. This figure shows age-associated change of intestinal flora by Dr. Mitsuoka, 1972. That is a very important achievement in intestinal microbial science. The ordinate indicates the number of bacteria per 1 gram feces. The largest number is 1 billion. That indicates multiply 10 to the 12th power. The largest groups are bacteria belonging to phylum Bacteroides, Eubacterium, and Streptomyces belonging to phylum Actinobacteria. Those are the largest group, 10 to the 12th power. 1 billion numbers in 1 gram. Following Bifidobacterium of phylum Actinobacteria, that number is 10 to the 10th power. The following is E. coli of phylum Gamma Proteobacteria, 10 to the 7th power. The next following is Lactobacillus phylum Actinobacteria, 10 to the 6th power. The lowest is Clostridium velci of phylum Firmicutes. The horizontal axis is age. Along to the ages, the key bacteria in gut is changing. From at the birth, newborn infant to weaning stage, there is a drastic change. That means a milk to food change. When get older, the declining of a bifidobacterium is observed when the aged people like me, more than 70 years old, the number of the bifidobacterium is decreasing. Upon this figure, introduction of lactobacillus and the bifidobacterium as a probiotic manner become an intestinal health practice. This slide just shows probiotics yogurt by lactic acid and bifidobacteria production. This slide shows a photo of shelves of yogurt by lactic acid and bifidobacteria in my neighbor supermarket. You can see in my neighbor supermarket, one big counter, big shelves are occupied by the lactic acid bacteria and the bifidobacteria yogurt. Next table shows major bacteria found in sewage compared to bacteria in human intestine. Once feces are discharged to sewer pipes, feces become main sewage component. Compared to bacteria in the human intestine, in sewage, phyla number 1, acidobacteria, phyla number 9, chloroflexi, phyla number 12, cyanobacteria, and phyla number 22, nitrosophila, emerged. This means a drastic environmental change happened for the bacteria. Partial aerobic condition occurs and biofilms are built up along inside of the sewage pipelines. Next table shows a comparison of major bacteria among activated sludge, sewage, and human intestines. Activated sludge, sewage, and human intestine. The activated sludge is a key bacterial consortium functioning in sewage treatment plants over the world. It has 1 century history that removes BOD, COD, nitrogen, phosphate, detergent, and other organic contaminants. Compared to the bacteria in sewage, 11 phyla are major bacteria groups in the activated sludge. Phyla number 16, LC microbia, phyla number 20, Gemma di monadites, and phyla number 23, Planktomycetes emerged. We can see a microbial ecological change among 3 situations. Among 3 situations, activated sludge, sewage, and human intestines. Now, this slide shows the idea of closing the loop, recycling organic waste in order to realize the idea of organic farming in order to practice the new technology of hyper-thermophilic composting is introduced in the next. This slide shows the model of Saga City in Japan wastewater treatment plant that accept 50,000 cubic meter per day of wastewater, that equivalent about 15,000 people. They treat sewage by the activated sludge method. They have digestion process, that is methane fermentation for sludge. After dewatering digested sludge, they compost by the hyper-thermophilic composting. They use 7 compartments for composting sludge for 7 weeks. They receive sludge and mix with a compost product that has a good seeding bacteria. After finishing compost, they sieve product, removing unnecessary things for agriculture. let's see the moving. This is a motion picture of the hyper-thermophilic composting for the garbage and the sewage. You can see the mixing garbage with the composted compost. It's a mixing. By loader, they are mixing. After piling up, they keep in a week. After a week, we can observe aerobic fermentation. And you see inside the pile, the high temperature produce. The temperature increase more than 100 degrees Celsius. He's picking up the product. You can see it's very dry powder, about 30% of moisture. Temperature of the composting pile increased more than 100 degrees Celsius. Finished compost become a seeding bacteria or the starting new compost. This slide shows a structure of a compartment where there are 3 side walls and the bottom are air diffusers. The right-hand side, you see piling up sludge about 2.8 meters. You see compartment by compartment. A pile of compost is destroyed and moved to the next compartment. After well mixed, a new pile is formed. Loaders are key machines for destroying, moving, and piling up a compost in the works. The consortium of heat-resistant bacteria is working in the composting. So that temperature goes up more than 100 degrees Celsius. This slide shows another composting place for a mixture of organic waste where they have a good record of temperature during 7 weeks. You can see left-hand corner, increasing the peak and declining altogether 7 weeks, declining temperature during each composting compartment. The highest temperature is more than 108 degrees at the second week. This figure tells killing microorganisms by temperature. The hyper-thermophilic compost increased temperature up to 100 degrees Celsius when temperature between 40 to 60 degrees. Fungi, nematodes, and plant seeds are killed. When temperature increases more, E. coli, norovirus, and the cucumber virus are killed. For the safety of agriculture, the hyper-thermophilic composting is a very useful technology. This slide shows bacterial change during hyper-thermophilic composting of sewage sludge. At the start, the water sludge contained less Firmicutes bacillus, and actinobacteria. Other bacteria occupied more. During the first and third weeks, bacteria species changing where Firmicutes bacillus becomes dominant. During fourth and seventh weeks, actinobacteria become dominant and the Firmicutes become second. This table shows major bacteria in the hyper-thermophilic compost of sewage sludge. In the hyper-thermophilic compost, together 13 phyla bacteria species emerged. Number 2, actinobacteria, and number 18 Firmicutes are major bacteria. Compared to sewage and human intestine, the hyper-thermophilic composts have unique ecology. Due to high temperature environment, it is natural to find phyla number 40, Deinococcus thermus, phyla number 29, Thermotogate, and phyla number 18, Firmicute thermus, Thermophilus, or Philus. Among them, phyla number 5, Bacteroides, phyla number 24, Proteobacteria become very important player with plant with Actinobacteria and Firmicutes. You saw this figure already. I am just remind your understanding of the figure. This bacteria, this shows a bacteria diversity of the rhizosphere. And then I introduce the PGPR bacteria, PGPR, plant growth promoting rhizobacteria, consist of the same groups of bacteria produced by the hyper-thermophilic composting. Now I'm going to change to the Africa business. NICO. I am a member of NICO, NGO based in Kyoto City, one of Japanese active international NGOs, which runs many projects Africa and Asia for about 40 years. They run comprehensive village development projects in Malawi during 2007 and 2014, and in Kenya from 2013 until now. NICO project consists of multi-components. African village need many aspects of development. NICO sets multi-components of their projects for development of a village. They always set water and sanitation a key component. Other components are shown in this slide. Many components are combined together. Among them, reforestation is also a key to provide wood energy for cooking food, accompanying ash for sanitation. This slide shows people to understand how to use the Ecosan toilet in Kenya. How to sit down over the toilet flat, urine collection, and defecation holes. After defecation, a boy showed that a handful of ash is thrown in the hole. After all, a boy washing hands with water. When ash is enough, ash is also used to clean hands with ash, that is alkali disinfection of hands. Protein, lipid, and carbohydrates are hydrolyzed in the high pH alkali condition, so that cell membranes of bacteria, viruses, and nematodes are injured. It is the principle of disinfection. A more clear picture, defecation hole covered by a lid, and in the center, urine collection, and a sack of ash. This slide shows that underneath an Ecosan toilet flat, there are two compartments of a feces box that are alternatively used. Once a component is full with feces and ash, the compartment is kept no touch for alkali fermentation until the other compartment gets full. The duration of one compartment is designed for a half year. In order to get a matured compost, it needs a half year for alkali fermentation. This slide showed the bacteria diversity for alkali compost of Ecosan human manure of boys' toilet. Boys' toilets of Bushangara Elementary School of Kakamega, Kenya. You can see actinobacteria and firmicutes and proteobacteria are major bacteria. There are other phyla species identified. This slide shows the bacteria diversity of alkali compost of Ecosan human manure of a family toilet in Kisumu, Kenya. Again, actinobacteria, firmicutes, and proteobacteria are major bacteria. Of course, there are other phyla species identified. Compared to the Bushangara human manure, proteobacteria occupied more. My understanding is this. This family use lesser ash after defecation. Again, I would like to recall your memory. The component, bacterial diversity of alkali ash component human manure is very similar to the PGPR diversity. This slide shows an agricultural test results of Ecosan human manure or maize yield. 4 sets of fertilizer condition. 1, Ecosan manure plus diluted urine. Number 2 set, diluted urine only. Number 3, chemical fertilizer. Number 4, no fertilizer. The result clearly show number 1 condition was the best. It gave more than customary application of chemical fertilizer. Only diluted urine application gave more than the chemical fertilizer. That means the soil is highly acidified and lost minerals for their long practice of agriculture. Their Kenyan soil are highly acidified. They lost minerals. Organic farming can enrich their poor soils. The bacteria diversity of soil can be enriched again. It is essential to keep sustainable use of the Ecosan toilet by the people. NICO provides Ecosan toilet with a self-help of the people participation. Your toilet, fermenting ownership, beneficially provides materials, beneficially helps the construction. You can maintain the toilet. Ecosan toilet is built with materials you can get in the local markets, not from Japan. A Japanese architect trains builders to construct it by themselves. Management by the Health Committee of Residents established the Management Committee of Hygiene and Sanitation in the village. NICO achieved missions in Malawi. They are continuing next project in other areas, including Kenya. 1,050 toilets have been constructed in Malawi. 220 toilets have been constructed in Kenya as of the 8th of December 2020. Checked the improvement of agricultural productivity by using Ecosan manure. Now we are proposing 1 million Ecosan toilets project. NICO will proceed Ecosan toilet project to the water catchment area of Lake Victoria. Like Kenya, Uganda, Tanzania, Rwanda, and Burundi contribute the improvement of water quality of Lake Victoria. They started Ecosan toilet project in India also. This slide shows annual washing style Ecosan toilets constructed in Myanmar. This style is welcome in many countries of South Asia, Southeast Asia, and Muslim areas of Africa. you can see the outside of the toilet. You see the human manure compartment and anal washed wastewater tank, and also inside anal washing flat, besides the defecation urination flat. And this is a design drawing of Ecosan toilet with anal washing style. In conclusion, in order to protect human and plant health, good soils must be sustained in agriculture. Composting technologies should be developed utilizing beneficial bacteria species. Hyper-Thermophilic composting is a promising technology. The ash alkali composting of human manure is also a promising technology for sanitation. Sanitation can be connected to organic farming. Beneficial bacteria should be explored for agriculture, such as Bacterial species, Actinomyces species, lactic acid bacteria species. The probiotic environmental agriculture has a hope. Thank you for your attention in my lecture.